Hey everyone. So this is my first video about something unrelated to cancer, but I think it has a lot to do with cancer therapy actually, because this video is about treatments in heart failure and heart disease that have everything to do with the cell cycle, with how cells progress through their cycle of reproduction and division, and how we can target that with precision medicine, with certain genetic and immunological techniques. And so without further ado, I'm going to get right into the video. Now, the first part of this video concerns a paper that kind of went unnoticed, unfortunately, in late November 2025. And it was a groundbreaking paper, I thought, because it really showed that heart cells, which we normally think of as the penultimate differentiated cell, I guess alongside neurons or brain cells, because they really don't de-differentiate. They're really hard to turn back into a fetal or a stem cell-like state. But this paper showed that's actually not true. And that's not true because of the main character of this story, which is cyclin A2 or CCNA2. It's a very important cell cycle protein. And so before discussing that, let's back up. So I said that heart cells really don't differentiate. What does that mean in the context of heart disease? Well, whenever a heart attack happens, whenever a clot bursts and breaks off into one of the major coronary vessels that feed the heart, that area of heart that is perfused by that blood vessel becomes ischemic. It's deprived of oxygen and blood, and it dies. And in response to that, whenever the heart goes back to normal after a heart attack, the remaining surviving cardiomyocytes, they can only respond to that injury by doing two things. They can only produce scars. For example, after a cut, you get a scab over your skin that is kind of scar or fibrotic collagen formation. Or they can hypertrophy, and that means those surviving cells really have to bulk up to replace the contractile function that was lost by those dying heart cells. Hypertrophy, of course, you may have heard in the context of skeletal muscle. After weightlifting, skeletal muscle tends to hypertrophy. But this is actually very bad in the context of the heart because, when the heart hypertrophies, the chambers of the heart can become, conducive to higher pressure and filling or less contractile function, or they can become electrically disorganized, which can cause arrhythmia, common cause of cardiac arrest. So this is obviously not a very optimal response after injury. And in fact, after the age of 10 in us humans, our cardiomyocytes are never even replaced. So by the age of 10 to 15 years old, we have all the heart cells we're ever going to have throughout the rest of our lives, even if we live up to the age of 90 or 100. So 80 years of those heart cells being vulnerable to any kind of damage. And this is a fixed amount. But there is a solution to this that I think we can kind of ascertain. In neonatal hearts, the hearts of neonates, of fetuses in the womb, and in some fish and amphibians, classically they undergo a process called epimorphic regeneration. So for example, in salamanders, as you can see in the top right corner there, if you cut off one of their limbs, they can actually regrow that back to the specific proportions of that amputated limb. This is called epimorphic regeneration. What it means is that the mature cells that are remaining after this traumatic event, they can de-differentiate to a certain extent. They can create a local progenitor cell population, and then they can regrow that limb very faithfully in that same morphology and structure. A very fascinating subset of regenerative medicine that we see in like amphibians and fish, even in neonates as well. And so this is a fascinating kind of feature but how can we apply it to heart medicine? Well, this is where CCNA2 enters, a very important cell cycle protein, as I mentioned. And CCNA2, it's been known as a candidate factor for certain regenerative techniques in cardiovascular medicine. In a model with pigs, they delivered CCNA2 after heart attacks. And this caused a 55% increase in total cell number in the regions around an infarct or the region around the heart attack zone. And this was without any hypertrophy or fibrosis, a really fascinating response. And so this brings us to the paper that was in 2025, Muhammad et al. What they did is fascinating. They introduced a human CCNA2 adenoviral vector into a population of adult human cardiomyocytes, that had been harvested from possibly, donors who had had heart attacks or heart failure. And from that, they saw an increase in a multiple degree of things. They saw increase or preservation in the cytokinetic index and proliferative index of these cardiomyocytes. So these cardiomyocytes, instead of being static and kind of non-growing, they actually start proliferating a lot, dividing a lot, having high mitotic index, which is fascinating. They also had an increase in their extracellular matrix and collagen synthesis, which is, of course, responsible for the holistic structure of organ tissues. the mold within which cells are kind of stacked up. They had an increase in fetal gene expression profile with a bunch of things that accelerate fetal cells through the cell cycle, which is very important. An increase in epithelial mesenchymal transition, which is very important because the heart is a mesenchymal tissue. And so epithelial to mesenchymal transition is crucial for regrowing this tissue faithfully. And then, very importantly, they actually saw a increase or a return in active calcium transients, which is how the heart really relies on contraction, how it beats in response to electrical rhythm. So in summary, not only were these cells growing and dividing like babies, not only were they doing that, they were also increasing their kind of structure and their electrical activity back to kind of a mature state. So the best of both worlds in a sense. And so this is a fascinating paper in these results. And just to put it up into an example, it's like you have a cookie and you baked it already, but this cookie crumbled or it became too overbaked and overcooked. And so somehow you turned it back into, a cookie dough ball that you can reshape back into a cookie that can grow, that can be molded into a better and a more optimal cookie. So this is kind of like how beautiful and how magical this transformation is with CCNA2. One caveat of this paper I do want to mention is that about 25 to 60% of human heart cells are binucleated, which actually does pose a challenge for stimulating increased proliferation because binucleated cells just don't proliferate as well as mononucleated cells. Just an interesting caveat that they discussed in their discussion section. And so just to recap, CCNA2 gene therapy could prove a very beneficial treatment that could prevent heart failure after these kind of ischemic events that we see. And you can see this here. They isolated and cultured these human cardiomyocytes that were supposedly terminally differentiated, unable to return back into the cell cycle at all. They introduced cyclin A2, and they saw all these things like reprogramming, like cell cycle reentry, a fetal-like transcriptome, cytokinesis, proliferation. Essentially, they turned these cells back into the playthings of, genetics of the playthings of proliferation, which is awesome. So just a sentence to wrap this up is that this is a plastic mesenchymal state that facilitates division while keeping the structural integrity of the heart intact. So basically the best of both worlds in regeneration, because the problem in regeneration is, of course we can turn cells back into a highly proliferative state, but then we could lose some integrity in some of that differentiated end organ function that we see with differentiated cells. But this kind of preserves both, and I think it's a really promising therapy in that regard. The next paper I want to discuss is actually derived from the Nobel Prize in Medicine that came out in 2025, and that was for the discovery of regulatory T cells. So I want to back up. The immune system is really, really powerful. It's one of the most unique systems in our body, but it's so powerful that it actually requires two kill switches to kind of prevent autoimmunity or self-reactive T cells from going out of control. The first kind of mechanism of this is the thymus. It's an organ that sits right underneath the sternum of our chest. Classically, in infants, it shows up like, very large, like a sailboat-like organ right in the middle of our chest, but it kind of shrinks over time as we lose the need for its function. And this causes central tolerance. But the one that they won a Nobel Prize for discovering was regulatory T cells. And these cause peripheral tolerance throughout the body. They can go everywhere. And they secrete things like IL-10. They cause contact inhibition for other T cells. They respond to IL-2, which is a kind of T cell growth factor. And so it's very important. But how is this important in heart disease? And, I have a picture of a T-Rex skull here because T-Rex sounds like T-Rex, just to help you remember that. Well, I think we have to recognize that one of the main contributors of heart and cardiovascular disease, heart attacks, strokes, pulmonary embolisms, is something called atherosclerosis. And atherosclerosis is essentially the buildup of fatty plaques within the walls of your blood vessels. And it's not just fat that builds up, it's also these inflammatory elements that build up in response to these oxidized lipoproteins that exist. These are things like monocytes, apolipoprotein B, which is a lipoprotein, also attracts a lot of these immune cells like flies to honey, basically. And in fact, trials show that when you deplete this inflammatory response with monoclonal antibodies like canakinumab in the CANTOS trial, it actually reduces the degree of major adverse cardiac events we see in patients that have had elevated inflammatory markers like CRP or C-reactive protein. The problem with this is if you dampen the immune system, you also predispose to microbial infection, which is, of course, why our immune system is so important in the first place. But this is where Tregs come in. In mice and humans, when we deplete regulatory T cells without depleting other types of T cells, we actually cause an increase in plaque instability and an increase in adverse cardiac events. And so this shows that regulatory T cells have a very important function in reducing the immune response, the pathological or the negative immune response that we see that contributes to atherosclerosis. And so this is a trial that came out, the Ivory trial. And what they did is that they specifically induced Treg cell expansion. And they did that, and that's without expressing or kind of inducing other inflammatory T cells. And they did this with something called low-dose aldesleukin, or IL-2. And that significantly reduces vascular inflammation, as we can see from a fluorodeoxyglucose positron emission tomography scan. And that's without increasing infection rates. So you can see the beauty of how selectively expanding regulatory T cells without expanding other T cells can really dampen the immune response that causes a vicious cycle of atherosclerosis and cardiovascular disease to accelerate. So I hope both of these papers really give you an insight into how cardiovascular medicine is really being transformed with our increased understanding of precision medicine, of genetics, and of immunology as well. And I think this is an exciting path for this field.